Last season, my neighbor planted tomatoes the traditional way, just tomatoes in neat rows with chemical pest control. I deployed the biological army strategy with 6 specific companions around my tomato beds. The difference was staggering. While he battled constant pest pressure and achieved modest yields, my tomato plants were surrounded by beneficial insects, showed minimal pest damage, and produced dramatically increased harvests. The secret wasn't luck or better soil. It was understanding how these 6 companions create a self-sustaining ecosystem that transforms tomato cultivation. Yet most gardeners continue using approaches that unknowingly sabotage their own success. The traditional approach creates this vulnerability through a fundamental design flaw that most gardeners never recognize. When you plant tomatoes in isolation, these neat rows and uniform spacing appear organized and efficient. The visual appeal of symmetrical tomato plantings reinforces the belief that this approach represents proper garden management. However, these isolated tomato beds function as concentrated broadcasting stations for chemical signals that attract destructive insects from across the landscape. Tomato plants naturally emit volatile organic compounds through their leaves, stems, and developing fruits. When grown in monoculture, these chemical emissions become concentrated and amplified, creating what agricultural scientists identify as a pest-magnet effect. The absence of biodiversity eliminates natural deterrents and beneficial insect habitat that would normally provide protection. Isolated tomato plants release concentrated chemical signatures that serve as location beacons for aphids, whiteflies, and hornworm moths. These chemical signals travel significant distances through air currents, effectively advertising the precise location of vulnerable tomato plants to pest populations throughout the surrounding area. Without companion plants to provide alternative chemical signatures or beneficial insect habitat, tomato volatiles dominate the local atmospheric chemistry. Aphids detect these concentrated signals from considerable distances, using specialized chemoreceptors to navigate directly to isolated tomato plantings where they encounter zero natural predators or deterrents. Research demonstrates that isolated tomato plants experience substantially higher pest pressure compared to those grown in polyculture systems. The concentrated nature of monoculture volatiles create pest aggregation effects that compound throughout the growing season. As initial pest populations establish on isolated plants, their feeding damage triggers additional chemical releases that attract even more destructive insects. This monoculture approach simultaneously eliminates natural pollinator habitat, forcing tomatoes to rely exclusively on wind pollination instead of the superior fruit set achieved through insect pollination. Tomato flowers require vibration and specific handling to release pollen effectively from their specialized structures. The absence of flowering companion plants eliminates nectar sources needed to attract and maintain beneficial pollinator populations. Native bees, hoverflies, and other pollinators require diverse floral resources throughout the growing season to establish breeding populations near garden areas. Monoculture tomato plantings provide no such resources, creating pollinator deserts where fruit set depends entirely on random air movement. This pollination deficiency significantly reduces both fruit quantity and quality compared to insect-pollinated tomato systems. The chemical pesticide cycle begins when desperate gardeners observe mounting pest pressure and respond with repeated spray applications. Initial pesticide treatments eliminate beneficial insects alongside target pests, creating ecological voids that actually increase vulnerability to future pest invasions. Subsequent pest generations develop resistance to applied chemicals, while beneficial predator populations remain suppressed. Each spray application reduces soil microbial diversity and eliminates parasitoid wasps that provide long-term pest control through biological mechanisms. These beneficial wasps lay eggs inside pest insects, creating self-sustaining population control systems that function throughout the growing season. Chemical applications disrupt these natural cycles, forcing gardeners into escalating pesticide schedules that become increasingly expensive and less effective over time. Pesticide applications also eliminate ground-dwelling beneficial arthropods that consume pest eggs and larvae before they reach damaging population levels. Predatory beetles, spiders, and other soil-inhabiting beneficial insects require sterile pesticide-free environments to maintain stable populations. Repeated chemical treatments create sterile soil ecosystems where pest species can reproduce reproduce without natural predation pressure. The fundamental problem lies in fighting against natural ecological principles instead of working with established biological systems. Tomatoes evolved within biodiverse ecosystems where multiple plant species provided chemical complexity, beneficial insect habitat, and pollinator resources essential for optimal tomato performance. Diverse chemical environments prevent pest aggregation while supporting beneficial insect populations that provide consistent pest control and pollination services. Monoculture systems eliminate these natural support mechanisms, forcing gardeners to substitute expensive external inputs for free ecological services. The solution requires understanding that tomatoes function as components of integrated biological systems rather than isolated agricultural crops. Companion plants activate natural defense mechanisms and productivity systems that have evolved over thousands of years. These biological partnerships provide services that no chemical substitute can replicate, including pest deterrence, beneficial insect recruitment, enhanced pollination, and soil ecosystem support. This fundamental shift from fighting nature to orchestrating ecological principles establishes the foundation for biological army approaches that work with natural systems. The key lies in deploying specific companion plants that recreate the chemical complexity and beneficial insect habitat found in natural ecosystems. These strategic partnerships transform your tomato bed into a sophisticated biological recruitment station. The first layer of your biological army consists of daisy family flowers that function as sophisticated nectar production facilities. Calendula and zinnias appear purely decorative when planted alongside tomatoes, with their bright orange, yellow, and multicolored blooms creating visually appealing garden displays. These composite flowers operate as complex biological machinery designed to attract, feed, and maintain beneficial insect populations throughout the entire growing season. Most gardeners scatter these flowers randomly throughout their garden spaces, treating them as aesthetic additions rather than strategic pest management tools. This random placement approach fails to maximize their pest-fighting potential and misses the critical proximity requirements that activate their protective mechanisms. The effectiveness of these nectar-rich companions depends entirely on precise positioning relative to tomato plants and understanding their specific biological functions within the integrated system. Calendula operates as a continuous nectar production facility throughout the growing season. Unlike many flowers that bloom in distinct phases or require specific temperature conditions, the composite flower structure provides concentrated nectar sources accessible to various beneficial insect species with different feeding requirements. These flower heads allow multiple insects to feed simultaneously without competition, creating efficient biological recruitment stations. The nectar attracts parasitoid wasps, microscopic beneficial insects that lay eggs directly inside aphids and caterpillar larvae. These wasps require consistent carbohydrate sources from nectar to fuel their search and destroy missions throughout tomato plantings. Female parasitoid wasps consume calendula nectar for energy, then systematically patrol nearby tomato plants searching for soft-bodied pest insects suitable for egg-laying hosts. Calendula serves a dual protective function by operating as a trap crop that draws aphids away from tomato plants when both options remain available within close proximity. This strategic diversion protects tomatoes from direct aphid infestation while concentrating pest populations on sacrificial plants where beneficial insects can efficiently locate and eliminate them. Zinnias extend the blooming period beyond calendula's natural cycle and attract different beneficial species through their distinct flower architecture and nectar composition. The flat, accessible flower heads of zinnias provide landing platforms for hoverflies, beneficial insects whose larvae function as voracious aphid consumption machines during their development stages. Adult hoverflies require zinnia nectar for reproduction and energy, while their offspring eliminate significant pest populations. Hoverfly larvae consume substantial quantities of aphids during their development cycle, with individual larvae processing hundreds of aphids before pupation. These beneficial insects remain active throughout warm weather periods, providing consistent pest control services as long as adult hoverflies have access to zinnia nectar sources. The continuous availability of nectar ensures that beneficial populations remain established within your tomato ecosystem, rather than dispersing to other locations. locations. The deployment strategy involves planting these nectar sources within close proximity to tomato plants to ensure beneficial insects patrol the immediate tomato zone rather than dispersing energy across larger areas. The optimal placement allows beneficial insects to efficiently transition between nectar feeding and hunting activities without excessive energy expenditure on travel between resources. When beneficial insects can access nectar within easy foraging distance of their hunting grounds, they establish patrol routes that provide consistent protection for tomato plants. Parasitoid wasps that feed on calendula nectar near tomato plants will systematically search those same tomato plants for pest insects throughout their active periods. This proximity effect concentrates beneficial insect activity exactly where pest protection provides maximum value. Tomato plots with strategically placed daisy family companions demonstrate measurably reduced pest insect populations and enhanced fruit set rates compared to isolated tomato plantings. The biological recruitment effect creates cascading benefits. As beneficial insect populations establish and reproduce within your garden ecosystem, their pest control effectiveness increases exponentially throughout the growing season. These flowers attract native bumblebees, which perform buzz pollination by gripping tomato flowers and vibrating their flight muscles at specific frequencies. This specialized pollination technique releases substantially more pollen from tomato anthers compared to wind pollination or casual flower visits by other insects. Bumblebee buzz pollination creates physical vibrations that dislodge pollen grains trapped within the tubular anther structures of tomato flowers. The buzzing frequency employed by bumblebees matches the natural resonance frequency of tomato flower structures, maximizing pollen release and ensuring comprehensive pollen transfer to stigma surfaces. This mechanical pollination process proves far superior to random wind movement or gentle flower handling by other pollinator species. Bumblebees require diverse nectar sources throughout their active season, making daisy family companions essential for maintaining these specialized pollinators near tomato plantings. Native bumblebees establish foraging territories around reliable nectar sources, creating consistent pollination services for nearby tomato plants throughout the flowering period. The presence of calendula and zinnia nectar ensures that bumblebees remain active within your garden ecosystem, rather than seeking resources in distant locations. This territorial behavior concentrates superior pollination services exactly where tomato plants require consistent flower visits for optimal fruit development. When positioned correctly, calendula and zinnias create a continuous recruitment system that builds beneficial insect populations throughout the growing season. The overlapping bloom periods ensure that nectar remains available during critical pest pressure periods and throughout tomato flowering phases. This biological foundation attracts and maintains the beneficial insect armies that provide both pest control and pollination enhancement services. However, attracting beneficial insects represents only the first layer of defensive measures. The second layer requires deploying aromatic plants that create invisible protective barriers around your tomato plants.
Basil and lavender deploy invisible chemical shields that disrupt pest navigation and communication systems around your tomatoes through sophisticated scent-based deterrence mechanisms. These aromatic herbs function as biological warfare stations, continuously broadcasting complex chemical signatures that interfere with pest sensory systems while maintaining beneficial insect activity levels. The volatile organic compounds released by these plants create atmospheric zones where destructive insects cannot effectively locate or communicate about tomato plant locations. The timing and placement of these aromatic barriers determines whether they provide comprehensive protection or accidentally repel beneficial insects needed for pollination services. Incorrect deployment can create chemical conflicts where aromatic compounds interfere with beneficial insect navigation or mask the nectar signals that attract parasitoid wasps and pollinators. Strategic placement requires understanding the specific effects these aromatic plants have on different insect species. Basil releases natural compounds that interfere with thrips and whitefly sensory systems through direct chemical disruption of their ability to locate tomato plants. These volatile compounds create sensory confusion that makes tomato plants essentially invisible to thrips and whiteflies during their host-seeking behavior. The scent specifically blocks the detection pathways that thrips use to locate tomato volatiles while disrupting whitefly communication systems used for aggregation and reproduction coordination. The molecular structure of basil's volatile compounds create competitive interference at pest insect insect receptor sites, preventing the detection of tomato-specific chemical signatures that normally guide feeding and egg-laying behaviors. Thrips rely on precise chemical gradient detection to navigate towards suitable host plants, and basil's emissions create false signals that misdirect their search patterns away from nearby tomato plants. Lavender produces natural compounds that create olfactory confusion for spider mites and flea beetles while simultaneously attracting beneficial predatory mites through selective chemical attraction mechanisms. These compounds interfere with spider mite pheromone communication systems, disrupting their ability to coordinate feeding sites and reproduction locations on tomato plants. The scent creates sensory overload conditions that prevent flea beetles from processing tomato plant location signals effectively. Predatory mites respond positively to lavender's emissions, using these chemical cues to locate areas with potential prey populations. These beneficial arthropods establish hunting territories around lavender plants, creating mobile pest control units that patrol nearby tomato plants for spider mites, thrips, and other small pest insects. The strategic placement involves interplanting basil among tomato plants to create continuous scent barriers that prevent pest movement between plants. This interplanting approach ensures overlapping volatile compound zones without creating excessive aromatic concentrations that might interfere with beneficial insect activity. Lavender positioning at corners create perimeter defense zones that intercept incoming pests while providing beneficial predatory mite habitat. Basil interplanting creates chemical corridors where pest insects experience continuous sensory disruption as they attempt to navigate between tomato plants. The integrated spacing prevents gaps in aromatic coverage while maintaining sufficient air circulation to prevent excessive humidity accumulation around tomato plants. Corner-positioned lavender functions as early warning stations that attract beneficial predatory species before pest populations establish on tomato plants. Research demonstrates that basil tomato intercropping systems produce measurable reductions in thrips damage while simultaneously increasing tomato fruit production through improved pollinator activity and enhanced plant vigor. The aromatic protection allows tomato plants to allocate energy toward fruit development rather than pest defense responses, resulting in larger fruit size and increased yield per plant. Beneficial insects attracted to basil flowers provide additional pollination services that complement the pest control benefits. The intercropping arrangement creates microenvironments where basil's flowering periods provide nectar resources for beneficial insects during critical tomato flowering phases. This synchronization ensures that parasitoid wasps and other beneficial species remain active within the tomato ecosystem throughout peak production periods. The dual-purpose nature of aromatic companions maximizes biological services while minimizing garden space requirements. These aromatic compounds mask the tomato-specific volatiles that normally guide hornworm moths to egg-laying sites on tomato leaves through chemical camouflage mechanisms. Hornworm moths use specialized chemoreceptors to detect tomato plant emissions from significant distances, following chemical gradients to locate suitable host sites. Basil and lavender volatiles create complex atmospheric chemistry that disrupts these navigation systems. The masking effect prevents hornworm moths from detecting tomato plants within aromatic protection zones, forcing them to search extensively without locating suitable hosts. This chemical camouflage proves particularly effective during peak hornworm flight periods, when moth populations actively seek tomato plants for reproduction. The continuous emission of aromatic compounds maintains protection throughout the entire growing season. When properly deployed, this aromatic layer creates a chemical fortress that protects tomatoes while enhancing rather than hindering beneficial insect activity through selective chemical effects. The sophisticated volatile compound interactions provide pest deterrence while maintaining or improving pollinator access to tomato flowers. This dual functionality ensures comprehensive protection without sacrificing the pollinator services essential for maximum fruit production. However, even the most sophisticated repellent system cannot eliminate all pest pressure. The third layer completes the biological defense network through an entirely different approach that appears to contradict everything we've established so far.
Nasturtiums and marigolds function as sacrificial decoys that lure pests away from tomatoes while providing additional beneficial insect habitat through sophisticated chemical attraction mechanisms. These plants appear to contradict the repellent strategy employed by aromatic herbs, but they actually complete the biological army by creating controlled pest management zones where destructive insects concentrate in predetermined locations. The trap crop approach operates on biological principles of preferential attraction, offering pest insects more appealing targets than your valuable tomato plants. Improper trap crop placement can backfire dramatically, concentrating pests near tomatoes instead of drawing them away to manageable locations where targeted intervention becomes possible. The success of trap cropping depends entirely on understanding pest movement patterns and deploying these sacrificial plants at strategic distances that intercept incoming pest populations before they reach tomato plants. Incorrect positioning transforms trap crops from protective barriers into pest aggregation sites that increase rather than reduce pressure on nearby tomatoes. Nasturtiums release natural compounds that aphids prefer over tomato-specific chemical attractants. These compounds create powerful chemical gradients that aphids detect from considerable distances, effectively overriding their natural attraction to tomato volatiles. Aphids possess specialized chemoreceptors that respond more strongly to nasturtium emissions than to the chemical signatures emitted by tomato plants. The chemical emissions function as biological magnets that redirect aphid flight patterns away from tomato beds toward strategically positioned nasturtium plantings. When aphids encounter these chemical signals during their host-seeking behavior, they abandon their search for tomato plants and concentrate on nasturtium foliage instead. This preferential attraction proves so effective that aphid populations establish breeding colonies on nasturtium plants rather than dispersing throughout your tomato ecosystem. Strategic positioning involves placing nasturtiums at bed perimeters to create pest interception zones that capture both soil-dwelling and aerial pest populations. The between-row positioning ensures that nasturtium chemical attractions reach sufficient intensity to override tomato plant signals without creating competitive growing conditions between the two plant species. Marigolds produce thiophines, specialized compounds that attract and trap nematodes within their root systems while simultaneously providing nectar for beneficial parasitoid wasps. Thiophines function as chemical lures that draw soil-dwelling nematodes away from tomato roots toward marigold root zones, where the compounds create hostile environments for nematode reproduction. This dual action mechanism protects tomato root systems while eliminating nematode populations that would otherwise cause substantial plant damage. The marigold root system becomes a biological trap where nematodes enter but cannot establish viable breeding populations due to the toxic effects of thiophene compounds. Nematodes attracted to marigold roots experience reproductive disruption and population decline, reducing the overall soil nematode burden throughout your growing area. This underground pest management occurs invisibly, while marigold flowers provide visible nectar resources for beneficial insects above ground. Marigolds also release pyrethrum compounds that repel the moths responsible for laying tomato hornworm eggs. These moths use specialized navigation systems to locate tomato plants for egg laying, but marigold emissions create chemical interference that disrupts their ability to detect suitable host plants. This additional protective mechanism prevents hornworm infestations before they begin, eliminating the need to manage established caterpillar populations later in the season. Marigold deployment strategy involves planting between tomato rows to create pest interception zones that capture both soil-dwelling and aerial pest populations. The between-row positioning ensures that marigold root systems intercept nematodes moving through soil toward tomato plants, while marigold flowers attract beneficial parasitoid wasps that patrol the entire growing area. This strategic arrangement maximizes both underground and above-ground pest management benefits. The trap crop system allows for targeted pest removal through simple leaf removal rather than treating entire tomato plants with broad-spectrum interventions. When nasturtium leaves become heavily infested with aphids, you can remove and destroy specific leaves or entire nasturtium plants without disrupting your tomato ecosystem. This targeted approach concentrates pest populations in disposable plant material that eliminates large numbers of destructive insects through minimal intervention. Heavily infested nasturtium leaves function as pest collection devices that simplify garden management while protecting beneficial insect populations throughout your tomato beds. The removal process eliminates concentrated pest populations before they can reproduce and spread to other garden areas. This mechanical pest control proves far more environmentally sustainable than chemical applications that kill beneficial insects alongside target pests. Research indicates that properly positioned trap crops reduce overall pest pressure on main crops while requiring minimal intervention compared to conventional pest management approaches. The trap crop strategy creates sustainable pest reduction through biological attraction rather than chemical deterrence or elimination. This approach maintains ecological balance while achieving substantial pest control benefits through natural behavioral manipulation. The biological effectiveness increases throughout the growing season as trap
crop chemical emissions intensify and pest populations become concentrated in predetermined management zones. Early season establishment allows trap crops to develop strong chemical attraction signatures before peak pest pressure periods begin. Mature trap crops provide maximum protection during critical tomato fruit development phases when pest damage creates the most significant yield losses. The trap crop layer completes a comprehensive system where pests are simultaneously repelled by aromatic plants, confused by chemical interference, and diverted towards sacrificial decoys in manageable locations. This three-pronged approach creates multiple barriers that prevent pest establishment on tomato plants through different biological mechanisms. The integration of repellent, confusing, and attractive strategies ensures comprehensive protection against diverse pest species with varying behavioral patterns. This three-layer approach creates a self-regulating ecosystem that intensifies in effectiveness throughout the growing season as each companion plant component matures and develops maximum biological activity levels. However, the pest control benefits represent only half of the biological army's potential impact on your tomato harvest.
The biological army system creates a pollination cascade that multiplies tomato fruit set far beyond what wind-pollinated monocultures achieve through sophisticated insect recruitment mechanisms. While pest control benefits provide visible evidence of companion planting effectiveness, the pollination enhancement operates as a hidden multiplier that dramatically increases harvest potential through biological services that remain largely invisible to casual observation. The nectar-rich companions establish pollinator highways that direct beneficial insects toward tomato flowers during critical pollination windows. Many gardeners focus exclusively on pest management aspects of companion planting and miss the substantial yield gains possible through optimized pollinator activity coordination. The pest control benefits capture immediate attention because damaged leaves and pest insects provide obvious visual evidence of problems and solutions. Pollination enhancement operates through subtle biological mechanisms that produce dramatic harvest increases without obvious day-to-day indicators of their effectiveness. The nectar-rich companions attract multiple pollinator species that visit tomato flowers throughout different times of day and varying weather conditions, creating comprehensive pollination coverage that wind pollination cannot match. Calendula and zinnia flowers provide landing platforms and nectar resources that support diverse pollinator communities with different activity patterns and environmental tolerances. These varied pollinator species ensure consistent flower visits during cool mornings, hot afternoons, and changing weather conditions throughout the growing season. Native bees, hoverflies, and other beneficial insects establish consistent foraging patterns around reliable nectar sources, creating predictable flight routes that include regular tomato flower visits. This consistent pollinator activity ensures that tomato plants receive regular attention throughout their flowering period, rather than depending on random pollinator encounters. Established pollinator presence provides services comparable to managed honeybee colonies without requiring hive maintenance or seasonal management. Bumblebees perform buzz pollination that releases substantially more pollen than wind or hand pollination methods, resulting in larger fruits with more seeds and improved shape characteristics. Buzz pollination involves bumblebees gripping tomato flowers and vibrating their flight muscles at specific frequencies that effectively dislodge pollen grains trapped within the tubular anthers. This specialized technique accesses pollen that wind and casual flower visits cannot reach effectively. The mechanical vibration employed during buzz pollination proves highly effective for releasing tomato pollen from its specialized anther structures. Tomato flowers evolved with anther designs that require specific handling to achieve maximum pollen release and transfer. Bumblebee buzz pollination provides exactly the mechanical stimulation needed to activate these specialized reproductive structures. Native sweat bees and hoverflies provide backup pollination services during cool mornings when bumblebees remain less active due to temperature constraints. These smaller pollinators tolerate lower temperatures and begin foraging activities earlier in the day than larger bee species. The diverse pollinator community ensures continuous pollination services regardless of daily temperature fluctuations or seasonal weather variations. Hoverflies contribute additional pollination benefits while their larvae provide pest control services, creating dual-purpose beneficial insects that enhance both pollination and pest management simultaneously. Adult hoverflies require nectar resources for reproduction and energy, while their offspring eliminate aphid populations during larval development stages. This biological integration maximizes the beneficial services provided by single insect species. Companion-planted tomato systems achieve substantially higher fruit set rates compared to isolated plantings through enhanced pollinator activity and improved flower visitation patterns. The increased pollinator diversity and activity levels translate directly into higher percentages of successfully pollinated flowers that develop into mature fruits. Enhanced pollination services prove particularly valuable during peak flowering periods when maximum fruit set determines final harvest yields. The continuous nectar supply keeps beneficial insects resident within your garden ecosystem rather than dispersing energy across larger foraging areas, ensuring consistent pollination services throughout the critical flowering period. Stable nectar resources eliminate the need for pollinators to search extensively for food sources, concentrating their activity within your tomato growing area. This biological containment effect maximizes the pollination services available to your tomato plants. Resident pollinator populations reproduce within your garden ecosystem, creating increasing beneficial insect density throughout the growing season. Successful reproduction requires adequate food resources and suitable habitat conditions that companion plantings provide through integrated nectar sources and diverse plant communities. The reproductive success of beneficial insects creates exponential increases in pollination services as the season progresses. The pollination multiplication effect explains how the biological army approach achieves substantial harvest increases through both pest reduction and enhanced fruit production mechanisms working simultaneously. Reduced pest pressure allows tomato plants to allocate energy toward fruit development rather than defense responses, while enhanced pollination ensures maximum conversion of flowers into mature fruits. These combined effects create multiplicative rather than additive. Superior pollination services produce larger individual fruits with improved seed development and enhanced shape characteristics that increase both harvest weight and fruit quality. Well-pollinated tomato fruits develop more uniform shapes and achieve larger sizes compared to inadequately pollinated fruits that remain small or develop irregular forms. The enhanced pollination also improves fruit flavor profiles and nutritional content through complete seed development processes. These biological services intensify as beneficial insect populations establish breeding territories and reproduce within your garden ecosystem. The increasing effectiveness creates momentum that builds throughout the growing season, establishing the foundation for long-term garden productivity that operates independently of external management inputs.
This momentum builds into a self-sustaining ecosystem advantage that distinguishes biological pest management from chemical approaches through fundamentally different operational principles. Unlike chemical treatments that lose effectiveness over time, the biological army system strengthens and improves throughout the growing season through regenerative biological processes. Chemical pesticides create a destructive cycle of resistance and repeated applications that escalates in both frequency and intensity as target pest populations adapt to applied compounds. The biological army approach operates through fundamentally different mechanisms that build beneficial momentum rather than depleting natural resources. Companion planting establishes self-reinforcing cycles where beneficial insect populations increase their pest control effectiveness through reproduction and territorial establishment. These biological systems function like compound interest. Small initial investments in beneficial habitat create exponential returns as beneficial populations establish breeding territories within your garden ecosystem. The initial weeks require patience as beneficial insect populations establish themselves within the companion planting system, creating a critical period where gardeners might abandon the approach before witnessing its full potential. This establishment phase demands faith in biological processes that operate on different timelines than chemical interventions. During this vulnerable period, beneficial insects must locate your garden, assess habitat quality, establish feeding territories, and begin reproductive cycles before their pest control services become apparent. Beneficial insects reproduce within your garden ecosystem, creating increasing populations of pest predators and parasites as the season progresses. Female parasitoid wasps that successfully complete their development cycles within pest insects immediately begin searching for new hosts upon emergence. The reproductive success of beneficial insects creates multiplicative effects, where single initial beneficial insects generate dozens of offspring throughout the growing season. These specialized beneficial insects possess remarkable host detection prediction abilities that improve through experience and population density effects. Higher beneficial insect populations create more intensive pest surveillance networks that detect and eliminate pest infestations before they reach damaging population levels. The host-parasite relationship creates biological feedback loops where increasing pest populations trigger proportional increases in beneficial parasite populations. The diverse plant community supports beneficial insects through multiple life stages, providing overwintering habitat and ensuring population continuity between growing seasons. Companion plants offer diverse resources, including nectar sources for adults, protective shelter during adverse weather, and overwintering sites that allow beneficial populations to survive seasonal transitions. Overwintering beneficial insects emerge in spring with established knowledge of your garden ecosystem and immediate access to habitat resources. Research demonstrates that established companion-planted systems achieve substantial pest control effectiveness by mid-season without requiring external inputs or management interventions. The biological momentum created by diverse beneficial populations reaches critical mass where pest suppression occurs automatically through natural predator-prey relationships. This autonomous pest control eliminates the labor requirements and material costs associated with repeated pesticide applications. Chemical pesticide effectiveness typically decreases throughout the season as pest populations develop resistance mechanisms and beneficial insects are eliminated from garden ecosystems. Repeated applications create selection pressure that favors pesticide-resistant genetic traits within pest populations while eliminating the beneficial species that provide natural population control services. This downward spiral requires escalating pesticide concentrations and application frequencies to maintain minimal effectiveness levels. The biological army creates a regenerative system that improves performance over time while reducing labor requirements and input costs through self-sustaining ecological processes. Established companion planting systems require minimal ongoing management once beneficial populations reach stable density levels and territorial establishment. The initial investment in companion plant establishment pays dividends throughout multiple growing seasons as beneficial insect communities persist and strengthen their pest control effectiveness. This self-sustaining advantage makes companion planting the superior long-term strategy for consistent, high-yield tomato production through biological rather than chemical pest management approaches. The regenerative nature of biological systems create increasing returns on investment, while chemical approaches generate decreasing returns and escalating costs over time. However, achieving this self-sustaining ecosystem advantage requires more than simply planting the right companions and the correct arrangements. The success of your biological army depends entirely on precise installation timing that coordinates beneficial insect establishment with tomato vulnerability periods.
The timing and sequence of companion plant installation determines whether your biological army provides maximum protection during critical tomato growth phases through synchronized deployment strategies. Random companion planting provides marginal benefits, but strategic timing creates coordinated protection that matches tomato vulnerability periods with beneficial insect population development. The deployment sequence functions like coordinated logistics. Each component must arrive when conditions optimize its effectiveness rather than when convenience dictates installation. Most gardeners plant everything simultaneously during weekend planting sessions, missing the opportunity to build beneficial insect populations before peak pest pressure begins. This simultaneous approach treats companion planting as static decoration rather than dynamic biological systems that require precise timing coordination. The rushed installation eliminates the population establishment periods that beneficial insects need to develop territorial behavior and reproductive cycles within your garden ecosystem. Calendula and marigolds should be planted several weeks before tomato transplants to establish nectar sources and begin beneficial insect recruitment processes. This advanced deployment allows these composite flowers to develop full blooming capacity before tomato plants arrive, creating established biological recruitment stations that attract parasitoid wasps, hoverflies, and predatory beetles. The early nectar availability ensures beneficial insects discover and colonize your garden space during their spring territory establishment behaviors. The pre-planting strategy transforms your garden into an active beneficial insect habitat before vulnerable tomato transplants require protection services. Calendula flowers develop multiple bloom cycles
cycles during this establishment period, creating continuous nectar production that sustains beneficial populations through varying weather conditions in seasonal transitions. Marigolds simultaneously establish root systems that begin producing compounds for underground nematode management. Early planted companions create biological momentum where beneficial insect populations establish breeding territories and begin reproductive cycles before major pest species emerge from overwintering sites. Parasitic wasps, attracted to early nectar sources, scout the surrounding area for suitable egg-laying sites, learning your garden's layout and establishing patrol routes that will later include tomato plants. This territorial establishment proves crucial for preventative rather than reactive pest management. Bezel installation coincides with tomato planting to provide immediate aromatic protection during the vulnerable transplant establishment period. The simultaneous planting ensures that volatile compound emissions begin immediately when tomato plants start releasing their own chemical signatures that attract pest insects. Basil's aromatic compounds create protective atmospheric zones around newly transplanted tomatoes during their most susceptible growth phase. Transplant shock makes tomato plants particularly vulnerable to pest attack during their first several weeks in garden locations. Root system establishment requires substantial plant energy that reduces natural defense compound production, creating windows of opportunity for pest insects seeking weakened host plants. Basil's immediate aromatic protection compensates for this temporary vulnerability by providing external chemical defenses. The concurrent planting strategy ensures aromatic barriers activate simultaneously with tomato volatile emissions that normally attract thrips, whiteflies, and other pest species. Young tomato plants produce different chemical signatures than mature plants, requiring specific protection strategies during early development phases. Basil provides comprehensive aromatic coverage during this critical establishment period. Lavender and nasturtiums are added 1 to 2 weeks after tomato establishment to complete the defensive perimeter without overwhelming young plants with excessive plant competition. This delayed deployment allows tomato root systems to establish territorial control over soil resources before introducing additional plant species that might compete for nutrients and water. The staggered installation prevents resource competition during vulnerable establishment periods. Lavender's aromatic emissions provide perimeter defense that intercept incoming pest populations before they reach established tomato plants. The delayed planting ensures lavender root systems develop in soil zones outside tomato root territories, eliminating competition while maximizing protective coverage. Nasturtium deployment creates trap crop functionality that diverts established pest populations away from strengthening tomato plants. This sequential approach ensures beneficial insects are present and actively patrolling before major pest species begin their reproductive cycles. Spring emergence patterns for most garden pests occur several weeks after beneficial insect activity begins, creating natural timing advantages when companion plants are deployed strategically. Early beneficial insect establishment creates population density effects that suppress pest colonization attempts. The deployment timeline also considers bloom succession, maintaining continuous nectar availability throughout the entire growing season. Different companion plants reach peak blooming periods at varying times, creating overlapping nectar production that sustains beneficial populations through seasonal transitions. Calendula provides early season nectar, while zinnias extend availability through late summer periods when other flowers decline. Sequential blooming patterns prevent beneficial insect population crashes that occur when nectar sources disappear suddenly. The planned succession ensures beneficial insects remain resident within your garden ecosystem rather than migrating to distant locations when local resources become unavailable. Continuous nectar availability maintains territorial behavior that concentrates beneficial services within your tomato growing areas. Proper timing transforms companion planting from static arrangements into dynamic defense systems that anticipate and prevent pest problems through biological population management. The strategic deployment creates cascading beneficial effects where early components establish foundation services that later components build upon and amplify. Each installation phase contributes specific biological functions that integrate with previously established systems. This strategic approach maximizes the biological army's effectiveness during critical fruit development periods when protection provides maximum harvest harvest value through synchronized biological services. However, without proper assessment methods, gardeners cannot determine whether their deployment strategy achieves the intended biological effectiveness.
Specific metrics reveal whether your companion planting system is achieving substantial harvest improvements and guide optimization decisions for maximum biological effectiveness. Without measurement protocols, gardeners cannot distinguish between marginal improvements and the dramatic gains possible with proper biological army deployment. The visual appeal of healthy-looking plants provides insufficient data to assess the complex biological interactions occurring within companion planting ecosystems. Many success indicators operate through subtle or delayed mechanisms, requiring knowledge of what to observe and when to assess system performance accurately. The most obvious signs of companion planting success, such as reduced visible pest damage, represent only surface-level indicators of much deeper biological transformations. Comprehensive assessment requires understanding the timing of beneficial insect population cycles, pollination enhancement effects, and soil ecosystem improvements that manifest over extended periods. Beneficial insect diversity serves as the primary indicator of biological army effectiveness, with successful systems supporting significantly higher beneficial species counts compared to monoculture planting. This dramatic increase in biodiversity indicates robust ecosystem functionality, where multiple beneficial species establish breeding territories and reproductive cycles within your garden environment. Species diversity assessment involves identifying parasitoid wasps, predatory beetles, hoverflies, native bees, lacewings, and other beneficial arthropods during systematic garden inspections. The beneficial insect counting process requires morning observations when most species exhibit peak activity levels and remain visible on flower surfaces or hunting territories. Different beneficial species operate on varying daily activity schedules, making comprehensive assessment dependent on multiple observations periods throughout daylight hours. Successful biological armies demonstrate consistent beneficial insect presence across different companion plant species and varying weather conditions. Pest damage assessment focuses on new growth and fruit development areas, where healthy systems demonstrate minimal damage throughout the growing season. This low damage threshold indicates effective biological pest suppression through natural predator-prey relationships rather than chemical intervention. Damage assessment requires examining tender growing tips, developing fruit surfaces, and young leaves where pest feeding typically concentrates during vulnerable plant development phases. The assessment protocol involves weekly inspections of predetermined plant sections, documenting pest feeding evidence, egg masses, and beneficial insect activity levels. Successful companion planting systems showed declining pest damage percentages as beneficial populations establish territorial control and reproductive momentum. Research indicates that properly implemented systems often demonstrate immediate, measurable pest reduction that provides early encouragement during the establishment period. Fruit set percentage comparison between biological army and control plants provides direct evidence of pollination enhancement effects achieved through beneficial insect recruitment. Companion planted systems typically achieve substantially higher fruit set rates compared to isolated plantings. This improvement demonstrates the pollination multiplication effect created by diverse nectar sources and resident pollinator populations. Fruit set assessment involved counting total flowers and successfully developing fruits during peak flowering periods. The measurement process requires marking individual flower clusters and tracking their development into mature fruits over multiple weeks. Enhanced pollination services become apparent through both increased fruit quantity and improved fruit quality characteristics, including size uniformity and shape development. Harvest weight and fruit count measurements demonstrate the cumulative impact of reduced pest pressure and enhanced pollination services working simultaneously throughout the growing season. These quantitative metrics provide concrete evidence of biological army effectiveness through measurable yield improvements. Weight measurements capture both quantity increases and individual fruit size improvements resulting from superior pollination and reduced plant stress. The measurement protocol involves weekly harvest documentation, including total fruit count, average fruit weight, and cumulative seasonal yields. Successful companion planting systems typically demonstrate substantial harvest increases compared to monoculture controls, with improvements becoming more pronounced as beneficial populations establish reproductive cycles. Late season measurements often reveal the most dramatic differences as biological momentum reaches peak effectiveness. Weekly beneficial insect counts during morning garden inspections track population establishment and reproductive success within your biological army system. Regular monitoring reveals whether beneficial species are successfully establishing breeding territories and producing offspring that remain resident within your garden ecosystem. Population tracking involves documenting both adult beneficial insects and evidence of their reproductive success through larval stages and egg deposits. The methodology requires systematic observation routes through companion plantings during optimal beneficial insect activity periods. Successful systems demonstrate increasing beneficial populations throughout the growing season as reproductive cycles produce multiple generations within established territories. Population stability indicates successful habitat provision and resource availability through integrated companion plant communities. These measurement protocols provide concrete evidence of your biological army's performance and identify opportunities for system refinement through data-driven optimization strategies. Documentation reveals which companion plant combinations produce maximum beneficial insect recruitment and which deployment strategies achieve superior pest suppression results. Measurement data guides future planting decisions and companion plant positioning for enhanced biological effectiveness. System refinement opportunities become apparent through comparative analysis of different garden sections with varying companion plant densities or species combinations. Areas showing superior beneficial insect diversity or reduced pest damage reveal successful biological strategies that can be replicated throughout expanded growing areas. Performance measurement identifies both successful elements worthy of expansion and underperforming components requiring modification or replacement. Successful deployment typically demonstrate measurable improvements within the first month to 6 weeks of establishment, with dramatic gains becoming evident by mid-season harvest periods. Early improvements appear through increased beneficial insect activity and reduced pest feeding damage. Mid-season measurements reveal the full potential of established biological systems operating at peak effectiveness levels. These measurement approaches transform companion planting from hopeful experimentation into systematic biological management that delivers consistent results through natural processes rather than external interventions.
The measurement data reveals how these biological systems transform traditional gardening from reactive pest management into proactive ecosystem orchestration. Your 6 companions, calendula, zinnias, basil, lavender, nasturtiums, and marigolds function as a complete ecosystem that provides services no chemical input can match. These integrated pest management systems create self-sustaining pollination enhancement and biological pest suppression through strategic deployment of beneficial insect habitat and natural deterrent mechanisms. The biological army approach represents a fundamental shift from fighting garden pests to orchestrating natural systems that make pest problems irrelevant while dramatically increasing productivity through established ecological principles. If you want to see exactly how I deploy these companions in different garden layouts and climates, let me know in the comments and I'll create a detailed implementation guide.